Hey guys, this video is going to introduce you to operons. This is technically the beginning of our next unit, right? Where we're going to talk about gene expression and the regulation of gene expression. Operons, let's talk about what they are. The first thing you're going to do is a couple questions up top on this doc. You can Google things to, if you need to remind yourself of some of these answers. I'm probably going to give away the answer of this in a second. What is an operon? An operon is a group of genes in bacteria that's under the control of one regulatory mechanism. So it's a group of genes. Genes make proteins. These genes will either make polypeptide subunits that combine to form one protein, or they'll make different proteins that act together in a function. But whatever the specifics are, the important thing is that the genes are expressed together as a group. Okay? So when you flip the on switch for an operon, suddenly you have the transcription and translation of several genes, right? So one regulatory mechanism, one on-off switch controlling this group of genes. Operons, I mentioned, are in bacteria. Operons have never been found in eukaryotic cells, right? Operons are specific to prokaryotes. That's why this doc is called Prokaryotic Regulation of Gene Expression, right? This is what you're going to see throughout this unit, whether it's gene expression, whether it's genomics, whether it's genetic engineering. What we know about DNA is typically discovered first in bacteria, and then we move on to eukaryotes. So that's how we're going to begin this unit, is how gene expression was first discovered, right? And it was the discovery of operons in bacteria. So an operon has a few parts to it. All the parts are the DNA. So we're just talking about areas of this strand of genetic material. There's an operator, a promoter, and then the genes of the operon. That's the operon itself. And then there's usually a regulatory gene. Okay, so let's take a look at this trip operon right here. Trip operon is a famous operon discovered in E. coli bacteria. And this is how it works. So first, we have the genes of the operon itself. You could call these things the structural genes. You could call them the functional genes too, but usually they're referred to as the structural genes of the operon, meaning these are the genes that actually make the polypeptides that structurally form the protein that we're talking about, right? So these, you can see there's 5 of them, and they each make a polypeptide subunit that forms an enzyme. And what does the enzyme do? The enzyme synthesizes the amino acid tryptophan. So this is kind of a little bit weird to think about, like a full protein making an amino acid. It's like a big brick building constructing a brick, right? But that's what this does. E. coli can synthesize the amino acid tryptophan. Honestly, just side note here, a lot of organisms can synthesize things that we can't, right? Like we synthesize our own vitamin D when we're in the sun, but most vitamins we need to get in our diet, there's really a lot of room for improvement with humans. But anyway, E. coli bacteria can make their own tryptophan. Now, that's what the genes of the operon do. Now, there's an operator. That's like an on-off switch. Right now, the operator does not have a repressor bound to it, so this operon is on, right? I can tell that. The reason I can tell it is because RNA polymerase here, which binds to the third part of the operon, the promoter, it has access to the operon. It can zip on down. Look at the direction of the red arrows. It can zip on down this operon and transcribe the mRNA that will go to the ribosome and make the polypeptide subunits that produce the enzyme. So right now, this enzyme is present in the bacteria. The trip operon is on. Right now, this is an example of a repressible operon. You got to know repressible and inducible operons. Repressible operons are ones that are on by default, but they can be turned off. Okay? So what type of gene would you want on all the time? You'd want a gene that makes something necessary on all the time. If the gene made something that was necessary only sometimes, you wouldn't want it on all the time, right? So the synthesis of tryptophan is important, right? Tryptophan is a needed amino acid for all kinds of protein synthesis. So the E. coli wants to have this operon on. Now, the only thing that would change that is if suddenly some food became present that had tryptophan in it, right? Like if suddenly, I mean, this is a gut bacteria, right? So if I'm in the gut of, say, a cow, and I'm E. coli, and I'm making my own tryptophan, if the cow suddenly eats a bunch of food, now suddenly